The crisis, it was a difficult moment for all of us, but one of the advantages is that we managed to work also in a different way, sometimes online, sometimes in a hybrid way, and this actually was good for all of us because we can manage to do more events in a way that is a bit more flexible compared to the in-person events. This webinar is organized in cooperation with the Coast Action CA 20121. The coast action is called From Bench to Bedside Transition for Pharmacological Regulation of NRF2 in Non-Communicable Diseases. The action is chaired by Professor Antonio Quadrado, who also will chair the session today. And I would like to thank him very much for this very nice cooperation. We are organizing several events together with a great help of Dr. Brigitta Buttari, that she's the main person actually behind the event today, and I would like to thank her very much. I also thank very much the two excellent speakers we have. They will be introduced by Antonio and Brigitta, and I want to thank also all of you, the participants, for being with us today, and I wish you a very pleasant and very fruitful meeting.
Okay, Professor Luciano Sasso, thank you very much. This is, again, a wonderful opportunity for everybody to learn from all these issues related to oxidative stress and also this fancy transcription factor called NRF2. And I can see among the audience many familiar names, people that I tend to believe now we are all colleagues and also friends, and I'm very happy for that. This is the first activity of the year for our cost action, also for the webinar series. And I think it is very exciting to have the commitment and the interest of both Professor Luciano Sasso at the Sapienza University and, of course, Brigitta Buttari, who is dealing mainly with the organization of this activity. I just want to say very, very quickly, just sort of a commercial, that we will have a meeting in Athens during February, dates 18, 19, and you are all welcome to connect online or even to come to Athens, although cost cannot afford to cover all the expenses, but at least you can connect online, and it will be related to the economical application of NRF2 research. It is an industry showcase. With this in mind, I wanted to go on again with the webinars. And hopefully, Brigitte, maybe you want now to introduce our first speaker. Thank you very much.
Hello, everyone. Thank you, Antonio and Luciano, for your introduction. It's a nice pleasure for me today to introduce Dr. Gloria Ritano. She's a researcher at the Department of Experimental Medicine at the Sapienza University. She was graduated in medicine and surgery and specialized in clinical pathology at Sapienza University. She moved after moving to Columbia University in New York for a brief period where she focused her research on the role of Notch4 in lymphangiogenic. She moved to Professor Maurizio Sorice lab. Maurizio Sorice have a consolidated, long-lasting collaboration. We started several years ago to investigate about the role of post-translation modification induced by oxidative stress on endogenous protein, and how this post-translation modification render this protein autoantigen in autoimmune disease, render this protein able to interact and activate the innate immune cells and contribute to the immunopathogenesis. So Dr. Gloria Ritano quindi start to collaborate with us, and her studies focusing on immunopathogenesis of autoimmune diseases. In particular, she is exploring the role of the regulation of autophagy and cell apoptosis and the functional role of lipid raft. She is engaged in the identification of new antigen and autoantibodies as possible biological markers. In addition, she is also involved in teaching general pathology at Sapienza University. So now I can leave the floor to Gloria for his talk.
Thank you, Brigitta. and can you see?
Yes.
Yes, we see. Maybe you can use full screen. Now we
Okay.
see better.
In this presentation, I will talk about the protective role of NRF2 pathway in antiphospholipid antibodies syndrome. The history of antiphospholipid syndrome dates back to 1906 when Wasserman first described a diagnostic test for syphilis. Meanwhile, a high number of patients tested positive for Wasserman reaction, and some of them have a prolonged clotting time in laboratory tests. This phenomenon was identified as lupus anticoagulant and was often associated with thrombosis, multiple abortions, and thrombocytopenia. After was demonstrated that the lupus anticoagulant and the positivity of Wasserman reaction was due to the presence of apparently of antiphospholipid antibodies directed against phospholipids. In 1983, Dr. Hughes described for the first time the syndrome of antiphospholipid antibodies characterized by multiple thrombosis, multiple abortions, cerebral disease, livedo reticularis, and lupus anticoagulant. One of the most serious manifestations of the syndrome is the cerebral thrombosis. And the syndrome has a wide clinical spectrum from asymptomatic to catastrophic forms. It can be classified in primary, more than 50%, and secondary when it is associated with systemic lupus erythematosus or more rarely other systemic connective diseases. To formulate a diagnosis of antiphospholipid syndrome, is needed at least one clinical criteria, such as vascular thrombosis and pregnancy morbidity, and one laboratory criteria. Laboratory criteria include lupus anticoagulant, anticardiolipin antibodies, and anti-beta-2 glycoprotein 1 antibodies. Despite the name of antiphospholipid antibody syndrome, many laboratories have identified a protein, the beta-2 glycoprotein 1, as the main antigen of the antiphospholipid antibodies. The beta-2 glycoprotein 1 can exist in different conformations, but the normal one is the ring conformation. In case of oxidative stress or post-translational modification of protein, the conformation changes and becomes similar to a hockey stick, and there is the exposure of the cryptic epitopes. The last domain can bind the phospholipids on the cell membrane, and the first two can bind the antiphospholipid antibodies. Furthermore, in recent years, with my research group, we dedicated to the study of the signaling pathways triggered by antiphospholipid antibodies involved in the pathogenesis of the syndrome. In previous studies, we demonstrated that the signaling pathways activated by antiphospholipid antibodies may involve lipid rafts, plasma membrane microdomains enriched in glycosphingolipid and cholesterol, with the production of anti-inflammatory cytokines and procoagulant factor, such as tissue factor, the main initiator of the coagulation cascade. Subsequently, with my research group, we demonstrated that antiphospholipid antibodies bind LRP6 with the activation of the signaling cascade through lipid rafts in endothelial cells. Like demonstrated by co-immunoprecipitation experiment with the LRP6 antibody, where a significant portion of beta-2 glycoprotein 1 was associated with the LRP6 after the antiphospholipid antibodies treatment. Western blot analysis showed that antiphospholipid antibodies induced the phosphorylation of LRP6, the phosphorylation of beta-catenin, and increased of expression tissue factor. The pre-incubation with methyl beta cyclodextrin, lipid rafts affecting agent, almost completely prevented this phosphorylation and this expression, suggesting the integrity of lipid rafts is essential for the signaling transduction. Furthermore, we elucidate the signaling transduction pathway triggered by antiphospholipid antibodies involved LRP8 in endothelial cells. Confocal microscopy demonstrated the association between GM1, a marker of lipid rafts, and LRP8 after antiphospholipid antibodies treatment. And this demonstrated that LRP8 is associated with lipid rafts. Antiphospholipid antibodies also induced LRP8 phosphorylation, DAB2 phosphorylation, and decreased the phosphorylation of endothelial nitric oxide synthase, supporting a fundamental role of lipid rafts in these pathways triggered by antiphospholipid antibodies. Furthermore, the inhibition of endothelial nitric oxide synthase with antiphospholipid antibodies increased the oxidative stress through the reduction of nitric oxide production. Indeed, modern theories have been suggested the two-hit hypothesis to explain the pathogenesis of antiphospholipid syndrome, where the first hit is represented by antiphospholipid antibodies and the second hit is represented by a triggering factor such as the oxidative stress. Oxidative stress arises from the imbalance between oxidants and antioxidants in favor of oxidants, and plays a role in the onset and progression of various diseases such as autoimmune disease. The role of oxidative stress in the pathogenesis of antiphospholipid syndrome has been widely demonstrated by experimental and clinical studies. It is considered the second hit contributing to the induction of the procoagulant phenotype in antiphospholipid syndrome. In response to oxidative stress, a variety of defensive molecular signaling pathways become activated. Among them, the transcription factor, nuclear factor, erythroid-2 related factor 2, or NRF2, It contributes to the anti-inflammatory and antioxidant processes. Furthermore, oxidative stress can trigger autoimmune disease by various mechanisms. One of these is the activation of the enzyme ADP ribose polymerase 1 that belongs to enzymes involved in DNA damage sensing and repair. PARP1 through NF-κB can induce the production of inflammatory cytokines, alarmins, prostaglandins, and modulate the inhibitory cytokines such as interleukin-10 and expression of FOXP3. So the oxidative stress plays a role in the breakdown of the immunological tolerance, contributing to the pathogenesis, pathological progression, and exacerbation of several autoimmune diseases. Antiphospholipid antibodies can trigger oxidative stress by various mechanisms, which can contribute to the thrombotic complications in patients with antiphospholipid syndrome. Indeed, oxidative stress is responsible for the formation of the oxidized LDL, and they bind beta-2 glycoprotein 1, forming a complex with proteinogenic properties. Patients with antiphospholipid antibodies syndrome have increased level of circulating oxidative stress markers such as 8-isoprostane, prostaglandin E2, C-reactive protein, and serum amyloid A. And moreover, a previous study demonstrated that antiphospholipid antibodies in monocytes of patients have increased the levels of peroxide, decreased the levels of nuclear NRF2, decreased the levels of intracellular glutathione with the alteration of mitochondrial membrane potential. And these dysfunctions were also induced in monocytes of healthy controls incubated with antiphospholipid antibodies. Antiphospholipid antibodies also promote the reduction of circulating antioxidant enzymes such as paranoxinase 1 and promote the alteration of nitric oxide production. Antiphospholipid antibodies also modulate the activity of the pro-oxidant enzymes of immune cells. Indeed, they activate the endosomal nicotinamide adenine dinucleotide phosphate oxidase with the induction of toll-like receptor 7 and toll-like receptor 8 in immune cells. And also antiphospholipid antibodies promote the activation of pro-inflammatory pathways in endothelial cells, platelets, monocytes, such as the activation of toll-like receptor for signaling with phosphorylation of IRAC, the phosphorylation of NF-κB, and the release of tissue factor and TNF-alpha. All these mechanisms contribute to the promotion of oxidative stress related to antiphospholipid syndrome. The alteration of oxidant and antioxidant in patients with antiphospholipid syndrome could represent new biomarkers for thrombosis risk. Animal studies and clinical studies demonstrated a reduced of total antioxidant capacity in patients with antiphospholipid syndrome. And furthermore, the increase of some oxidant markers such as F2 isoproteins in patients with antiphospholipid syndrome demonstrated to have a predictive role in cardiovascular events. Moreover, oxidative stress can induce the post-translational modification of proteins, and they represent a potential trigger for various autoimmune diseases by generating new antigenic epitopes with the consequent loss of self-tolerance. The non-post-translational modification of proteins in antiphospholipid syndrome involved mainly the beta-2 glycoprotein 1. Beta-2 glycoprotein 1 mainly be subjected to oxidation, glycosylation, phosphorylation, and acetylation. And in a recent study, we identified the beta-2 glycoprotein 1 carbamylated as a new antigenic target in antiphospholipid syndrome. Beta-2 glycoprotein 1 carbamylated is able to activate dendritic cells, and the serological results showed that a significant part of patients with antiphospholipid syndrome were positive for anti-beta-2 glycoprotein 1 carbamylated antibodies. The research of these antibodies has proven to be particularly useful for the diagnosis in patients so-called seronegative who have the signs symptoms of the syndrome but are persistently negative on laboratory tests. For this reason, this test has an international patent. Many papers demonstrated the interplay among oxidative stress, inflammation, and NRF2 pathways in antiphospholipid syndrome. Antiphospholipid syndrome is characterized by uncontrolled inflammation and oxidative stress. Recently, in animal models, was demonstrated that trichloroSN exposure that increased oxidative stress accelerated autoimmune responses by the dysregulation of toll-like receptor signaling and NRF2 signaling. But the antioxidant supplementation, such as sulforaphane, clearly restored the NRF2 signaling in animals exposed to trichloroethane. In addition, NRF2 signaling pathway upregulates various cytoprotective enzymes and regulates about 1% of human genes by the antioxidant response element OR. The NRF2R signaling pathway may be considered the primary pathway for intracellular redox balance. NRF2 is physiologically located in cytoplasm and anchored to KEAP1, forming a complex KEAP1 NRF2, and NA3 ubiquitin ligase promotes the ubiquitin-mediated degradation of NRF2 with the maintenance of the basal steady-state level of activity. Under oxidative stress, NRF2 dissociates from KEAP1 and translocates to the nucleus. The target genes comprise phase 1 and phase 2 detoxification enzymes, transporters, growth factors, and also genes that increase anti-inflammatory cytokines such as interleukin-10. In previous paper was demonstrated that in patients with antiphospholipid syndrome, the NRF2 activity was repressed, suggesting a defective signaling of NRF2. The cytoprotective role of NRF2 was demonstrated in animal models. Indeed, mice lacking NRF2 developed several autoimmune-mediated lesions similar to those of human systemic lupus erythematosus, the development of autoantibodies, premature death. Moreover, NRF2 had interplay with toll-like receptor signaling, showing that NRF2 protects against injury related to oxidative stress, modulating at toll-like receptor 7. From these premises, the pharmacological regulation of NRF2 in antiphospholipid syndrome could have a cytoprotective role, preventing tissue injury related to oxidative stress, and could represent an emerging treatment strategy in antiphospholipid syndrome. But however, antiphospholipid syndrome is a complex pathological process in which various multiple signaling pathways are involved in immunopathogenesis. The knowledge of the interplay between the inflammatory pathways and the antioxidant responsive pathways contribute to a better comprehension of the immunopathogenesis of antiphospholipid syndrome, driving towards a personalized medicine. Nowadays, there is an increasing interest in nutritional interventions because of their potential role in preventing tissue injury related to uncontrolled inflammation and oxidative stress. Recently, Nocella and co-authors provided an overview of the use of the antioxidant supplementation such as omega-3 polyunsaturated fatty acids, vitamins, coenzyme Q10, and polyphenols to prevent oxidative stress and atherothrombotic complication in patients with antiphospholipid syndrome. However, the lack of data from sufficient randomized controlled trials should slow down the process of antioxidant supplementation, becoming a potential management option in antiphospholipid syndrome. Many natural compounds have been reported to promote the activation of NRF2 signaling pathway, regulating redox homeostasis, inflammation, leading to cell survival and tissue repair. And more than 1,000 genes have an antioxidant response element in their promoters and can be activated by NRF2 during oxidative stress. Many experimental studies have shown the protective role of NRF2, thus making this pathway a valuable therapeutic target in chronic diseases marked by low-grade chronic inflammation and oxidative stress. Indeed, a recent paper demonstrated that artemisinin decreased the inflammation and autoantibodies production in kidney with the activation of NRF2 signaling. Another paper demonstrated that taxifolin, a flavonoid found in apples and onions, decreased the autoantibodies production and large vein thrombosis in animal models of antiphospholipid syndrome. Another paper demonstrated that monocytes treated with antiphospholipid antibodies had increased inflammatory, increased the activation of inflammatory pathways, but the pre-incubation with the coenzyme Q10 decreased for example, the activation MAP kinase pathway. Nowadays, several bioactive compounds have been identified as NRF2 activators, and they mediate protection against injury related to oxidative stress and inflammation, thus representing new therapeutic strategies in patients with antiphospholipid syndrome. The comprehension of the interplay between different anti-inflammatory pathways and the antioxidant responsive pathways implicated in the immunopathogenesis of antiphospholipid syndrome could lead more and more towards a personalized medicine. Thank you for attention.
Okay, thank you very much. Please, could you stop sharing? Thank you very much for this wonderful talk. I think we learned a lot about this autoimmune disease, quite interesting. We will have the questions at the end of the webinar. So we will now move to the next speaker. And it is for me a great honor to introduce Professor Gerasimos Sikiotis, Jerry, for our, is much shorter and easier to pronounce. Thanks, Jerry. So Jerry is a physician scientist specialized in clinical and basic endocrinology with a particular focus on thyroid physiology and thyroid diseases, including thyroid cancer. Jerry graduated in medicine and obtained his PhD degree at the University of Patras in Greece. His postdoctoral training in internal medicine and in the endocrinology was conducted in Patras and also at the University of Rochester Medical Center in Rochester and at the Massachusetts General Hospital in Boston. He worked for Novartis Institute for Biomedical Research in Cambridge, Massachusetts, and since 2015, he is responsible for the thyroid clinic at the endocrinology service of Bauth University Hospital Center in Lausanne. His basic research focuses on the roles of cellular antioxidant response systems in thyroid physiology and pathophysiology, and his clinical research focuses on the needs of patients with thyroid diseases, including quality of life among thyroid cancer patients and survivors. So, Jerry, thank you very much, and please go ahead.
Thank you very much, Antonio, and many thanks also to Vicita and Luciano for this kind invitation. I will now share my screen. And today we'll discuss the thyroid gland more in its basic research aspects and related to autoimmunity and how it also translates to the humans. Because I don't expect the audience to be very familiar with the thyroid, I will first give a brief overview of the thyroid and its physiology, and especially the role of oxidation in the synthesis of thyroid hormones, and some basics on thyroid autoimmunity. After that, I will present some of our studies on NRF2 in thyroid physiology, which gave us some hints for possible links to autoimmunity. And then I will discuss the studies we have done and we are doing to characterize the links between NRF2 and thyroid autoimmunity in more detail. And I will finish with some take-home messages for a non-specialist audience on thyroid, including when to think about the thyroid in your own projects. So the thyroid hormones act on all cells of the body. They influence basically everything: cell differentiation during development, growth, and metabolism throughout life. and they are required for the well-being and proper functioning of all tissues. The thyroid, as I expect everyone knows, is located here in the lower neck, in front of the trachea and the larynx. And what is more relevant to us is the anatomical units of the thyroid gland, which are these spherical structures called follicles, where we have the epithelial cells in the periphery and basically a liquid in the center. These are the cells that produce the thyroid hormones. And they do this in a way which is briefly described as follows. Let's suppose we have here a slice through a thyroid follicle, and this is one epithelial cell. This cell is like a machine that is designed to do two main things: to take up iodine from the blood. This iodine comes from what we eat, and to take it inside and then move it through specific protein transporters to the other side, close to the surface of the cell here. And the other thing it is made to do is to synthesize a high molecular weight protein called thyroglobulin. Half of the weight of a normal thyroid is thyroglobulin protein. And this thyroglobulin is also exported here close to the surface. And this is where the oxidations take place. They are catalyzed by an enzyme that is called thyroid peroxidase. We have three sequential oxidation reactions. Iodine is oxidized at the atomic level, and then atoms of iodine are connected to tyrosine residues of thyroglobulin. We can have tyrosine residues with 2 atoms of iodine or with 1. And in the next oxidation reaction, these are brought together. And if we have 2 plus 2, we end up with this hormone. And if we have 2 plus 1, we end up with this hormone. This is called T4 or thyroxine, and this is called T3 or triiodothyronine. These are now still on the molecule of thyroglobulin, which is then taken up by the same cell or a neighboring cell, doesn't matter. And this endosome is fused to lysosomes, and there is a proteolysis process that breaks down thyroglobulin and releases the thyroid hormones on the other side of the cell into the systemic circulation. So to kind of simplify this, we can say that we have iodine, and this is used to iodinate thyroglobulin. And this is done through oxidation reactions that are triggered by hydrogen peroxide, that the thyroid gland has dedicated enzymes to actively generate hydrogen peroxide. And this leads in the end to the formation of the thyroid hormones. Because oxidative stress is important for the synthesis of thyroid hormones, it is not surprising that it has been shown before that a certain level of oxidation is required for thyroid cell proliferation and function. So a very reducive environment is not proper for these processes. And since the thyroid needs oxidation, it also is not surprising that it has to be able to tolerate it. So it was also shown that it has increased tolerance for oxidative stress. And further, in certain conditions that are pathological, where there is either not enough iodine or the thyroid, for various reasons, becomes big, we call this goiter, so goitrogenesis, there is even more oxidative stress in the thyroid. And then this triggers an antioxidant response. When we started to work on the NRF2 system in the thyroid, we were interested in questions like how the thyroid cells sense oxidative stress, how they launch these antioxidant responses, and whether and how the redox status of the thyrocyte is coupled to the actual iodine metabolism and the synthesis machinery for the hormones. And we speculated that this might have something to do with the NRF2 system. Now I will mention some general aspects of thyroid autoimmunity, and then I will bring these two together. I will speak about, there are different forms of thyroid autoimmunity. The most common one, which I will talk about today, is called Hashimoto's thyroiditis. It is named after a Japanese physician that described it more than 100 years ago. It is very common in the population. It's about 5% of the population, 3 to 4 times more common in women, like most autoimmune diseases. And I think we are 60-something people on this webinar, so for sure a few of us have Hashimoto's thyroiditis or are going to develop it in the future. It is also called chronic autoimmune thyroiditis, or chronic lymphocytic thyroiditis. Why? Because there is a strong infiltration of the thyroid gland by lymphocytes. We see here the follicles, normal follicles, but most of this thyroid gland has been taken up by infiltration by the immune system. In contrast to other forms of thyroiditis, there is this chronic inflammation, so chronic autoimmune thyroiditis, without acute destruction of the thyroid follicles. There is destruction, and it leads to loss of thyroid function, but this is generally very progressive. It is also the most common cause of hypothyroidism in areas where there is enough iodine in the diet, like most of the developed countries in our days. And interestingly, we heard about from Gloria about how oxidative stress can unmask or create autoantigens. And here, similarly, excessive iodine exposure can lead to too much or improper iodination of thyroglobulin, which then becomes an autoantigen for the immune system. The infiltration by lymphocytes, we can also see it on the ultrasound when we image the thyroid. I've put here the outline of the thyroid in this patient. This is the trachea. And these more dark areas are where there is a lot of infiltration by the immune system, that we see here. In addition to this infiltration by the lymphocytes, there are also antibodies, both in the thyroid and circulating in the blood, where we can measure them also for diagnostic purposes. And these antibodies, they target the enzyme that catalyzes the hormone synthesis reaction, the protein that is the precursor of thyroid hormones, the receptor for the pituitary hormone that regulates all of thyroid function, and others. So we have both a cellular and a humoral autoimmunity. Hashimoto's thyroiditis develops or follows certain stages. There is a component of a genetic predisposition, and there are SNPs that have emerged from both candidate gene and genome-wide association studies that predispose to Hashimoto's. Some of them are in the HLA system, others are in inflammatory mediators, and others are in proteostatic mediators. I will mention some of them in next slides. Then, together with probably some environmental exposures, as I mentioned, for example, exposure to high iodine, the next step is to develop the autoimmunity, and clinically we detect this by the measurement of thyroid autoantibodies, but still with normal thyroid function. Next, as progressively the thyroid follicles are destroyed, the first thing that happens is that the system tries to equilibrate. So the pituitary will secrete more of this hormone. It's called thyroid stimulating hormone that tries to keep the thyroid going. Initially, it will manage to do so. So we have normal T3, T4, but increased TSH, and we call this compensated or subclinical hypothyroidism. And when eventually the thyroid is destroyed to a degree where it cannot produce sufficient hormone, then we have, in addition, low T3 and T4 in the circulation. We call this overt hypothyroidism. These patients, they need to be treated with thyroid hormone. Some of these patients may also need to be treated with thyroid hormone, for example, women who are pregnant. So in some of our, I will now start to present my work and then try to bring these two topics together. In one of our studies we did, we used mice and we exposed them to high amounts of iodine, and we were interested in the responses of their thyroids and the role of NRF2 in these mice. So we had wild-type mice and NRF2 knockout mice, and either on normal conditions or they were drinking a high amount of iodine in the water. So technically easy experiment to do. We treat them for 1 week and then we sacrifice them. We take blood, we take thyroid, and some other tissues, and from the thyroid we get mRNA and protein for molecular analysis. What we found in this experiment is that, well, of course, there are several genes going up, going down in the different genotypes. One thing that was quite clear is that the iodine exposure activates the NRF2 response. You can see several typical NRF2 target genes like NTO1, their induction level in wild-type mice with iodine versus controls. and much lower levels in the knockout animals with iodine versus controls. And overall, the whole pathway was activated in an NRF2 dependent manner in response to iodine. What does it do? Why is it activated? Because it seems that it ameliorates the immune, inflammatory, and fibrotic response that takes place in response to iodine. So you can see here many immune, inflammatory, and fibrotic responses, which were overall increased in the knockout animals when NRF2 is not present. So it means NRF2 has a protective role against the responses to iodine. In a related work using the same experimental design, we went into more detail in some of the mechanisms involved in these processes. So same experimental design, and here you can see, for example, the antioxidant response with NTO1 being induced in the wild-type animals with iodine and having low levels and no induction in the knockouts, and similar patterns for other antioxidant genes with those two with known important roles in the thyroid from previous work by others. And as you would expect from an antioxidant protector, the NRF2 response protects the thyroid against the protein and lipid oxidation that happens in response to iodine, specifically in the knockout animals. So these are here pooled protein samples from different groups of animals with these from these groups. And the only group that has excess protein oxidation, as you can see, is the knockouts when they are treated with iodine. And similarly, the only mice that have increased levels of this oxidized lipid for hydroxynonenal in the follicles are the knockout animals exposed to iodine. The arrows point to fat around the thyroid as opposed to control. So specifically when NRF2 is missing, then we have excess oxidation in response to iodine. Perhaps even more interestingly, we found that NRF2 has a direct relationship with thyroglobulin. This is independent of iodine. So in the knockout animals, thyroglobulin mRNA levels are lower. And on the western also, we can see lower on the Kumasi gel, and also on the western, we can see lower levels of thyroglobulin in the knockout animals versus the controls. This is independent of iodine. And by looking at the regulatory elements of thyroglobulin, we could identify two antioxidant response elements, and we could show that both of them are transcriptionally active and respond to NRF2 activators, and both of them bind the NRF2. I will not walk you through this data. This is reported in assays, and this is chip sequencing in the interest of time. But we clearly characterized both of these as functional AREs. So a direct relationship between NRF2 and the expression of the main gene involved, or one of the main genes involved in thyroid hormone synthesis. Conversely, and even more interestingly, we found that NRF2 regulates negatively the levels of iodinated thyroglobulin. So this is total thyroglobulin. These are individual mice in each lane. And here we're looking at iodinated thyroglobulin. So in the knockouts, there is clearly more iodinated thyroglobulin. And when the mice are treated with iodine, instead of iodinated thyroglobulin decreasing like it does in the wild type, it increases even further. So we don't know yet what is the mechanism of this phenomenon. We suspect it may have to do with the degree of oxidative stress in these thyroids. We are testing that in parallel. But I remind you, when you have too much or not proper iodination of thyroglobulin, this is something that may be linked to autoimmunity. And these animals here are black 6 mice, which are resistant to autoimmune diseases in general. They are prone to metabolic disease, as you may know. So it was not a good model for us to test directly this linker, but I will come back towards the end to how we are doing this currently. So these were hints for links between NRF2, iodine exposure, and autoimmunity in the thyroid. Another interesting aspect is that we know that NRF2 protects different organs, and we know this thanks to studies with these mice that have shown that these mice are sensitive to pretty much anything you can do to them that involves oxidative stress. And here are some examples of disease models. And these disease models also include experimentally induced autoinflammatory and autoimmune conditions. For example, experimental colitis, experimental myocarditis, arthritis, and so on. And if you do nothing to these mice, and you just watch them, if the NR2 mutation is in an autoimmune permissive background, the mice will develop age-related multi-organ autoimmunity. I should say it has been shown for several organs. The thyroid has not been looked at specifically. So for these reasons, we speculated that NRF2 may also be involved in thyroid autoimmunity directly. And we did a genetic study in humans to test this hypothesis. In this study, we collaborated with colleagues from Portugal who had previously done some work in their country with a cohort of patients and controls. They had about 1,000 participants. About half of them had Hashimoto's thyroiditis at the final stage. So this patient had overt disease. They needed thyroid hormone treatment. And about half of them were unrelated controls that had normal thyroid function. The Portuguese colleagues had studied functional polymorphism in the promoter of a gene encoding a selenoprotein. Selenium is very important for the thyroid function, and it is sometimes used as a treatment in early stages of thyroid autoimmunity. And there are many selenoproteins that are important for different aspects of thyroid homeostasis. So it made sense to study this gene. And in this candidate gene association studies, they could show that presence of the minor allele in both males and females increased the risk of Hashimoto's, more so in males than in females, but at least twofold compared to non-carriers. So then, knowing that the NRF2 promoter also has functional polymorphisms, functional meaning that the minor allele decreases the expression of the gene. And these have been associated with different diseases. We thought to check whether there is a genetic interaction between the two. So we genotyped, in collaboration, of course, the NRF2 polymorphisms in the promoter of NRF2 in these same individuals, 1,000 individuals. And when we looked at the data, depending on how many minor alleles, so alleles with decreased transcription, were present in these different individuals, only those that had minor alleles for both selenoprotein and NRF2 had increased risk for Hashimoto's. So none of the two genes individually was important when the data were looked at in combination. And it didn't matter how many minor alleles were present for NRF2, at least one was enough to increase the risk. In parallel, we did some molecular studies in humans and mice, and we found that in patients with Hashimoto's thyroiditis, these patients had been operated for other reasons, because we don't operate Hashimoto's thyroiditis. But if patients are operated for something else, often it may be that they also have thyroiditis. So we can look at that. And we found with the immunohistochemistry that there was a decreased expression of the selenoprotein S protein in Hashimoto's patients versus controls. And when we looked at our NRF2 knockout mice, we found a decreased expression of selenoprotein S protein in thyroid follicular cells. This is summarized here, whereas there was no difference in the liver. So this seemed to be specific to the thyroid. we checked whether selenoprotein S is a transcriptional gene of NRF2, like thyroglobulin I showed before, and with different experiments that I will not walk you through, it was not the case. So no difference between NRF2 knockouts or KEAP1 knockout cells. These are thyroid follicular cells in culture. It's a cell line that we have from rats, and we have made with CRISPR knockouts for NRF2 and KEAP1, so we can use it as a model. No evidence for direct transcriptional regulation. Conversely now, when we targeted selenoprotein in the cells, we used here siRNAs to decrease its expression levels. In combination, you can see 3 siRNAs very nicely silenced selenoprotein S. And when we did this, we had lower expression of NRF2 target genes at basal conditions when we stimulated with sulforaphan, an NRF2 activator, or when we stimulated genetically in the KEAP1 knockout cells. Always the SI for selenoprotein S decreased the NRF2 target gene expression, and it also decreased the viability of the cells. So these data together, they imply that there is a bidirectional positive crosstalk between NRF2 and selenoprotein S, not at the transcriptional level, but the post-transcriptional level. And at the same time, there is this genetic interaction between the two. And you can see that these two pathways do very similar things. Both of them suppress inflammatory cytokines. NRF2 is more specific for oxidative stress, and selenoprotein S for ER stress. And together, we believe these two synergize to promote or to determine the sensitivity or resistance to Hashimoto's. Now, I will discuss something conceptually a little different, and we will come back to autoimmunity again. We were wondering if when NRF2 activators are used for other indications, like other autoimmune diseases or non-autoimmune diseases, are they safe for the thyroid? So one way to approach this is to know or to ask what is the effect of genetic activation of NRF2 on the thyroid, because the pharmacological effects may be similar or not. And also what is known from preclinical and clinical studies of NRF2 activators about the thyroid, if anything. In humans, already 10 years ago, there was a very nice, very convincing case report with one family from Japan, in which a loss of function heterozygous frame shift mutation in KEAP1 co-segregated perfectly with enlarged thyroid, goiter. So this would argue that NRF2 activation in the thyroid causes goiter. Of course, with an N equals 1, you can never be sure, because it may be some linked mutation or something else, or some non-coding gene or something else. So we looked at it specifically in the mice that have flox sites around the KEAP1 gene. These flox sites, without any CRE, decrease the expression of KEAP1 and increase the activity of NRF2. And we found that these mice have enlarged thyroids. These are the mice at 1 year of age of these mice. We can see clearly the difference. This difference is also starts to become obvious already at 3 months, and the goiter increases with age. You can see here the structure of the follicles. It looks normal, but they're big in the KEAP1 mice compared to wild-type mice. These mice also had a tendency for decreased thyroid function. What I mentioned before as the subclinical hypothyroidism, where the pituitary hormone is increased, but the peripheral hormones are still normal. In these mice, in recent work, we exposed them to high iodine. What I showed you before for NRF2 mice, we did the same for the KEAP1 knockdown mice with activated NRF2. And we found interesting aspects from this experiment. I'm showing you here only the graphical abstract. We found that the inflammatory response that was increased in the NRF2 knockout here is decreased. This may be expected, so one would argue that it would be protected from autoimmunity and predisposition. We found some aspects that are specific to how the thyroid handles iodine that I will not get into. And we also found that in this 1 week experiment, give them iodine in the water for 1 week, the KEAP1 knockdown mice had an enhanced cell growth and proliferation signaling. So then the hypothesis is that under normal iodine conditions, but in long-term, let's say, observation or maintenance, this would lead to the goiter phenotype. What do we know from clinical studies? Tom Kensler, that many of you know, has done clinical trials with sulforaphane or sulforaphane-producing beverages in China for chemoprevention purposes. So we collaborated with Tom and took advantage of samples that were available from participants in these trials to see if there was any evidence for effects on the thyroid, either thyroid hormones or thyroid autoimmunity. So these were patients that had been drinking either placebo or a sulforaphane generating beverage for 3 months. And overall, there were no differences in thyroid function or thyroglobulin, circulating thyroglobulin levels. And there was no difference in the percentage of patients that could be, or the number of patients that could be classified as having a mild hypothyroidism, or the number of patients that had in their blood antibodies for thyroid proteins like TPO, TC, as I mentioned before. So there was, it seems to be safe for the thyroid, at least from this data. And we are currently analyzing data from an experiment that we did in mice, where we gave them a sulforaphane-producing beverage. We put it in their water and they drank it, and it had no effect on thyroid weight, function, or immune infiltration over 3 months. We know this is stable because when we leave it on the bench for several days, then we use it on our thyrocyte cell line, it can activate NRF2 signaling like sulforaphane does. So it is stable, and you can give it in the water in this way. We did not want to do gavage or parenteral administration because those can have effects on the levels of thyroid hormones, even just from the stress that it causes to the animal. So we had no evidence for any toxic effects or effects on thyroid autoimmunity with this treatment. There was a case report with another drug. This was for sulforaphane. This is dimethyl fumarate. Dimethyl fumarate is used for psoriasis, and it's also used for multiple sclerosis. And it activates NRF2. It also has immune effects, and its efficacy in multiple sclerosis or other diseases may be more related to those immune effects than to NRF2 activation. But in any case, it is an NRF2 activator. And there was a case report that described the exacerbation of Hashimoto's thyroiditis in a patient that was treated with dimethyl fumarate for their multiple sclerosis. Let's say it was not very convincing, the data, when I looked at this. So then we went to our hospital, and the cohort of patients that are treated with dimethyl fumarate or had been treated with dimethyl fumarate for multiple sclerosis, and out of 163 patients, had very few with any evidence for altered thyroid function. Only 2 of them during or after dimethyl fumarate. One had a mild transient hypothyroidism during treatment. One had autoimmune hyperthyroidism after this treatment, and when they were on another disease-modifying autoimmune working drug. So could not find any convincing signal of thyroid toxicity in patients on DMF. So I would say the evidence to date points to the safety of these drugs for the thyroid. And what we are currently doing, and I don't have any data to show for this, it's an experiment that we have been preparing for a while. We're going to test directly the role of NRF2, its genetic activation or inactivation in a mouse model of thyroid autoimmunity. This is called the NOD-H2H4 mice. They develop autoimmune thyroiditis. They develop inflammation of the salivary glands and also the lacrimal glands that produce the tears. It's a polygenic model. They are derived from the NOD mice, non-obese diabetic mice that develop type 1 diabetes. These mice, they don't develop type 1 diabetes, but they develop age-related autoimmune thyroiditis, celladenitis, dacryadenitis. And what's interesting is that when you give them extra iodine, thyroiditis develops faster, it is more prevalent, it affects more animals, and it is more severe. And here are examples of mice that were for several months on normal diet or increasing concentrations of extra iodine. And you can see the degree of immune infiltration in their thyroids. And it goes hand in hand with developing thyroid autoantibodies that can be measured in the serum of these animals. So, after 2 years of backcrossing, we will soon have our colonies with NRF2 knockout mice in the NRF2 knockout mutation in the node H2H4 background and the KEAP1 hypomorphic mutation in the node H2H4 background. We will do the experiment giving them high iodine concentrations to test directly whether NRF2 is protective and whether NRF2 activation is further protective or not in these animals. So this could also educate us about whether pharmacological activation of NRF2 in the thyroid, if we manage to do it, might also be helpful in this context. So in conclusion, I've shown you several of the many roles of NRF2 in the thyroid, in antioxidant defense, production of thyroglobulin, iodination of thyroglobulin, autoimmune regulation, and some other aspects in response to excess iodine. Please remember, genetic activation of NRF2 can lead to goiter, and pharmacological activation, according to the evidence to date, seems to be safe. I would also add that this should be tested specifically for each and every NRF2 activator that is used in the clinic. And some take-home messages for non-thyroidologists. Oxidation is central to thyroid function. NRF2 has these pleiotropic roles I just mentioned. Genetic activation and goiter in both mice and humans. And NRF2 is involved in thyroid autoimmunity in humans. We will test this more directly and mechanistically in mice. And finally, we want to think about the thyroid in your projects. If you're studying metabolism and you are finding differences between your groups of interest, please consider whether this may be due to altered thyroid hormone function or status. And if you're using NRF2 modulating compounds or drugs in animal models or clinical trials, it's important to test for thyroidal safety, I would say. This is my team. These are our collaborators, our funding sources. Big thank you to the Bandbed Thyroid Cost Action, led by Antonio, and I'll be happy to take any questions. Thank you very much.
Okay, Jerry, thank you very much. I think it was really a wonderful, very comprehensive and explicative talk about the thyroid physiology and also the role of oxidative stress. I think we can now start a round of questions. Maybe you can put your questions in the chat and either Brigitta or I will read them. And while you decide about, we already have one here. Actually, we have more. Yeah, we have more than one. Maybe Brigitte, Brigitta, would you like to read this one from Elisabetta?
Yes, I can read it. A question for Dr. Ritano. Did you evaluate the role of autophagy in nerve activation and or autophagy-induced drugs in APS? A question for the doxycyclitis. Okay, we can read the question for Jerry after. Quindi the first question is for Gloria. Quindi Gloria, did you evaluate the role of autophagy in nerve activation on autophagy-induced drugs in APS?
Thank you, Brigitta, for this question, this beautiful question. Autophagy and NRF2 pathways are measuring intracellular defense systems that combat oxidative damage and maintain homeostasis. However, the interplay between these two pathways remains largely unknown. And but one paper or some papers precisely demonstrated that P62 phosphorylated that is an autophagy activator and is necessary for autophagic degradation of polyubiquitinated proteins, dramatically enhances its affinity for KEAP1, which induces KEAP1 to release NRF2. So there is the permanent degradation of KEAP1, and eventually NRF2 can translocates to the nucleus and activate its target genes. And the other, there is an inducing drugs, inducing autophagy drug that can be used in the antiphospholipid syndrome. It is the rapamycin. Rapamycin is an inductor of autophagy because it is an inhibitor of mTOR. mTOR is involved in the endothelial proliferation related to nephropathy in antiphospholipid syndrome. So rapamycin can be useful, and there are also other drugs.
What do you think about metformin?
Metformin is an FDA-approved anti-diabetic agent, and it is used for the management of type 2 diabetes. And the metformin also can activate autophagy, inhibiting mTOR, and could be useful, but it needed some randomized clinical trials also, such as other supplements.
Okay, another question for you, and then we can pass to Jerry. Okay, in the light from Rita Businaro, in the light of the signaling pathway you described, which compound do you suggest to use for the treatment of antiphospholipid syndrome?
Yes, if you I presented some our papers related to the lipid rafts involving in the signaling pathways of antiphospholipid syndrome. Indeed, drugs that affect lipid rafts can be used, such as simvastatin or statins, in general statins, and also cyclodextrin. Simvastatin derived from the Aspergillus terreus, so it's completely natural, and this kind of drugs.
Ok, thank you. We can move to Jerry. Quindi a question, from Elisabetta Profumo from ESS. Okay, data demonstrate that E3 ubiquitin ligase SMARF1 upregulate NRF2, protect against endoplasmic reticular stress, and promote glioblastoma cell survival. Did you evaluate its possible role in cancer and or in thyroid autoimmunity?
Yes, thank you for this question. I have not studied SMARF1. I'm not very familiar with it, I must confess. We have studied NRF2 in cancer, and this is actually the first, I didn't present this because it was not directly relevant for this topic. This is actually the first work we published 10 years ago, where we showed that NRF2 is activated in the most common form of thyroid cancer. It's called papillary thyroid carcinoma. In contrast to other cancers, most of the time there are no mutations in KEAP1. So it's likely activated by mutations either in other genes or or by non-IPAP genetic mechanisms. Other groups have also shown later that NRF2 is activated in this form of thyroid cancer and in other forms of thyroid cancer. So there is some interest in developing NRF2 inhibitors or potentially using NRF2 inhibitors when they are developed for other indications, to use them also for forms of thyroid cancer that are resistant to standard treatment, which is surgery and radioactive iodine. For SMERF1 specifically, I cannot comment because I don't know much about it.
Okay, another question is from Marina Marini for Jerry. Do you think that NRF2 may have a role in the regulation of thyroid activity that does not require iron binding and regulation of iron-dependent genes?
Yeah, it's a good question. I don't have the answer. I would say, okay, so we have some effects that are direct transcriptional effects. We know it regulates the transcription of thyroglobulin. This we know, black and white. But for some other effects, we don't know the mechanism, or it does not seem to be transcriptional, directly transcriptional. For example, the iodination of thyroglobulin that is increased in the absence of NRF2. But it may be transcriptional in an indirect way, because if you don't have the transcription of the antioxidant genes, you may have more oxidative stress in the thyroid, and this may lead to more iodination of thyroglobulin. This is something we are testing now directly by giving N-acetylcysteine to the mice to decrease the level of oxidation in the thyroid in both wild type and knockout, to which we give iodine at the same time, and to test whether the mechanism is at that level. So yeah, it's a good question. I don't have a direct answer about other mechanisms, transcriptional ones.
Okay, another question for you from Rita Businaro. Do you think that it may be possible to prevent or treat thyroid autoimmunity by targeting NRF2? What about ozone therapy? I
have no idea about ozone therapy. About the possibility to prevent or treat thyroid autoimmunity by targeting NRF2, yes, this is also my question that I would like to answer. I would say that if we are, first of all, we need some drug or some compound or some antioxidant natural substance or something that can actually get to the thyroid and activate NRF2 in the thyroid. And as of now, we don't have good evidence for any compound getting to the thyroid in meaningful concentrations to do that. And we would need it to be able to do that and to not be toxic to other tissues at the same time. We would also need it to be safe for the thyroid. And we have these individuals who developed these hereditary goiters because they had genetic activation of NRF2. They had genetic activation of NRF2 in the whole body. Like the mice, they had genetic activation of NRF2 in the whole body. But they came to medical attention because of the thyroid. So it means maybe the thyroid is super sensitive to NRF2 modulation. So maybe we can use it as a target, maybe not. It's a very good question. I would say, in general, for potential treatments, preventive or therapeutic for thyroid autoimmunity, the understanding is that they may have some effect if they are done early, in the phase where the inflammation has started or is about to start. There may be some destruction of the thyroid tissue, but not a lot. If we are in a late phase where the thyroid has been destroyed, it's atrophic, the patient needs to take thyroid hormone. This is not something that any antioxidant or other anti-inflammatory or immune-modifying treatment would be able to treat. About ozone therapy, I'm happy to learn, so please contact me and we can happy to discuss.
Jerry, a question for me. How the diet may influence the effect on NRF2 in the thyroid?
Yes, this is a very good point. We know from what I showed you, the very high amounts of iodine clearly have effects on the thyroid. What we have observed is that even within the physiological range of iodine intake, there may be effects. So the diets that our animals eat in the different facilities and different countries, different universities, the content of iodine and also of selenium in the diet is very different. It is considered physiological, but it may be 5 times, 10 times different between different commercial chow. So we have observed that this can affect the phenotypes, like the goiter phenotype I showed you. We have some observations that this phenotype depends on the amount of iodine in the diet. And currently, we are testing this in a formal, properly controlled manner. So I expect that this will have a big relevance. And this is what we're doing currently. Thank you.
Okay, thank you. No other question I see in the chat, and if
Brigitte, if I may, maybe I could also ask a couple of things to Jerry. So I am indeed curious about the effect that you said that apparently there is not evidence of target engagement of NRF2 activation in the thyroids, either in the Chinese study with sulforaphane or in the studies in multiple sclerosis with Tecfidera, with dimethyl fumarate, or even in the mice that you mentioned that you were using with sulforaphane. So this is a very critical point, because either we don't get enough to activate NRF2, or maybe based on the genetic activation that you got with the hypomorphic KEAP1, maybe too much of NRF2 may be a problem. So what do you think?
Yes.
So the question is, how can we balance the amount of NRF2 activation that we need? Because with the current drugs, we don't see activation, and in the genetic side, we see too much activation.
Yes, I don't have the answer. I think I would like to. I would like in my dreams. Whenever a clinical or preclinical study is done that uses NRF2 activators, the thyroid function would be analyzed. This is easier to do in humans. Of course, it's maybe more costly to do in humans. And of course, if we think it may be for safety purposes, clinical investigators may not be eager to go looking for toxicity unless the authorities specifically ask them to. So in preclinical studies, it's not so difficult. It's quite easy, actually. You can just take you don't need to measure the hormones in the blood. You don't need to touch the thyroid. You should just take the pituitary of the animals and measure TSH mRNA expression in the pituitary. If you see any differences, that means that thyroid function is altered. So it means the drug is getting to the thyroid and it's changing thyroid function. So in my dreams also, any colleague who's working with NRF2 related models, genetic or pharmacological, would also take out the pituitary and measure TSH mRNA in that tissue. Yeah, and I would be eager to. So I wrote to the investigator of ameloxolone study and suggested to measure TSH in the blood of these patients. I didn't get an answer. That's life.
Now, this is because most likely they are doing it by themselves.
No, I think they don't want to know about any potential toxicity. That's what I think. But we'll see.
Okay. Is there any other question? Okay.
May I just put a little comment? I want to thank also Gloria for her nice presentation. I learned a lot about the phospholipid syndrome. And by the way, 20% of these patients with antiphospholipid syndrome also have Hashimoto's thyroiditis. So I think it was good connection of the
Yes,
topics.
indeed. It's a secondary antiphospholipid syndrome. It could be considered the secondary for this association.
Okay, so just let me remind you before we close, let me remind you that in February 18, 19, we will have a meeting, online meeting for most of you that you can easily join us in Athens about the pharmacology of NRF2. And we will see how small and big companies are going after NRF2 related therapeutics. So just if any of you is not getting the information and wants to join, just send me an email or to Brigitta or someone in the action, and you will immediately receive the link to join us. Okay, Brigitta, would you like to close, please? You are mute.
Okay. Eccomi. Okay, thank you so much, the speaker, for their interesting talk and clear answers. Thank you everyone for participating to this webinar. I hope this webinar has been informative and helpful, so share it with your colleagues and students. I want also to remind you that the recorded webinars are always available on the website, antiox.it, or in our cost action web page. So share this information. I'm looking forward to keeping in touch for next event. The next webinar is scheduled for the spring, and the topics should be oxidative stress and aging. So I want to say hello and have a nice weekend.
Thank you.
Okay.
Bye-bye.
Bye-bye.
Okay, so thank you to everybody, and bye-bye.
Bye.
See you again in a few weeks.
Bye-bye.
Bye.
Bye bye.